Hello and welcome everyone. On behalf of the Biochemical Society and Portland Press, we are really pleased to welcome you to today's webinar. This is part of a dedicated Biochemistry Focus Early Career Researcher webinar series. The topics in this series include a large number of different areas in the molecular biosciences and also practical sessions to support career development. Each of the webinars will give you the opportunity to ask questions via text, and we also welcome suggestions for future topics and speakers to feature in this series. So please see the website for more details. I am Dani Ungar. I'm a professor at the University of York. My interest is very much in glycobiology and cell biology, and I'm sharing the chairing of this webinar with Karen Polizzi, who I'm going to pass over to now.
Hello everyone, I'm Karen Polizzi. I'm a professor of biotechnology at Imperial College London in chemical engineering, and my research in this area is interested in the overproduction of recombinant therapeutic glycoproteins with the right glycoforms. So I also wanted to take the time to mention that Dani and I are coordinating an upcoming Biochemical Society meeting that's very closely related to today's webinar topic, and you might be interested in attending this. It's called From Golgi Organization to Glycan Function, and it will be taking place in Brighton on the 5th to the 7th of April in 2022. And this meeting is really about bringing glycoscientists together with cell biologists and fostering new ideas and collaborations between both disciplines. So there's a range of topics on the program, from traditional cell biology, glycobiology, through to analytics and synthetic carbohydrate chemistry, all the way to systems level studies. And so we really would like to get these communities to come together and have joined up thinking about this topic. There are still oral communication slots available at the meeting, and we would like all the attendees to consider to register, particularly early stage career researchers. So you can submit either a poster abstract for consideration or an oral communication. And the abstract deadline is the 7th of February, so please do check the website for more details and make sure you don't miss an opportunity to submit the abstract by the deadline. The conference in April will also be live streamed, so you're welcome to register to join us remotely if you prefer.
Right, so today's webinar is titled Developments in Glycobiology, and it is part of our dedicated early career research program of webinars, as I mentioned. And today we will welcome 3 early career research members of the Society, and they will share their work in the field of glycobiology with the biosciences community. So we will hear from Ben Schumann at the Crick Institute and Imperial College, from Fiona Cuskin at the University of Newcastle, and from Salvatore Santamaria from Imperial College. So I will then first just introduce Ben Schumann, and once he has given his presentation, I will introduce the next speakers. If you have questions to the speakers, these will be discussed at the end, but please feel free to add your questions during the talks. Please just type them into the question box as shown on the image on the screen, and we will then try to answer as many of these questions as our time allows at the end. So our first speaker is Dr. Ben Schumann from the Crick Institute and Imperial College London. Ben has completed his undergraduate studies in Tübingen, Germany, and his PhD at the Max Planck Institute of Colloids. And for this work, he obtained the prestigious Otto Hahn Medal of the Max Planck Society in 2017. He then moved to Stanford University to work with Caroline Bertozzi, who has really pioneered the use of chemical biology for the study of glycans and glycan functions. And Ben has then started his own group at the Crick Institute in 2018 to really take the chemical biology approach to the study of mammalian glycans. And Ben is going to now talk to us about his research. Please, on chemical precision tools to study the glycoproteome.
Thanks so much, Danny. Sorry, I just needed to unmute myself. Thank you so much for the introduction, for the opportunity to speak here. There's a pointer. We are the Chemical Glycobiology Lab at the Francis Crick Institute, and we are, as Dani said, really interested in making chemical tools to understand how glycosylation works. And I thought, kind of as the first speaker here, just to kind of give everyone a little bit of a primer, maybe to the people who are not yet glycobiologists but are getting into this field, which is a cool rabbit hole to be in, so definitely stay in that field. Just to show how glycans can diversify the proteome, right? And this is taken from a paper in 1996, human CD2, which is a small protein. It has no relevance for the rest of this talk, although it has huge relevance for health. And what we would normally see, we would be looking at a protein in PDB, in the protein data bank, is kind of the structure here, which usually omits glycans. But since this was taken from an NMR structure, we can see a single N glycan on this protein, and we realize kind of how much the glycan adds to this protein, how much it diversifies the plasticity of the protein. And just kind of zooming into cell surface glycosylation, we realize that there's a lot happening with different monosaccharides represented by these different schemes of colorful circles and squares and so on. That again diversify the proteome, and sometimes the addition or ablation of a single monosaccharide can make a huge difference on whether two proteins interact, for example. But understanding glycosylation is not really easy because glycans are not directly encoded in the genome, but instead are made in the combinatorial interplay of 250 glycosyltransferases. So if we want to understand how glycosylation works, we need to understand glycosyltransferases. And there have been huge efforts in doing this from many great players in the field. But chemistry can play its part there. And what we've been able to do, and what the field has been able to do for the last 2 decades or so, maybe a little bit more, was to take particular sugars such as N-acetyl-galactosamine or GalNAc, yellow square, as the glycobiologists would say. Structure is not really important for this talk. To take GalNAc and make a chemically modified version of it that is clickable, so it has a functional group that allows for modification with a fluorophore or biotin after incorporation into the glycoproteome. So the sugar is taken up by a cell, incorporated into the proteome, and then you can use Crick chemistry to introduce a fluorophore or biotin. And this can be an enabling technology because it allows for enrichment of glycoproteins and then mass spectrometry, or for detection in the context of imaging or the CRISPR screens or more fancy instrumentation in biology. But the downside has always been that these clickable sugars are not necessarily specific for individual glycosyltransferase enzymes. They have been rather blunt tools to kind of visualize the whole of the glycocalyx, for example, which is in itself really important. But they couldn't tell us much about the activity of individual glycosyltransferases. And so we are setting out to change that. And what we want to do, and this started in a collaborative project with the Bertozzi lab, who have largely pioneered this, was to kind of see the tree among the forest, right? See individual glycans or individual substrates for glycosyltransferases, protein substrates for glycosyltransferases, by introducing chemical handles. And this is a cover image we had in Molecular Cell in 2020, which was made by my student Mia Zolhan. And so the way this works is, and the way this has been pioneered in the Bertozzi lab, and we have kind of adapted this in our lab, was to use a strategy called bump and hole engineering, which again had been around for a while, but has never been kind of amenable to use on glycosyltransferase until recently. And what you do is you take an enzyme that binds a UDP sugar, in this case UDP GalNAc. Again, the structure isn't too important here, but what's important is that this is an activated version of a sugar that is used by glycosyltransferases. And so this wild-type enzyme binds this UDP sugar and transfers the sugar to a peptide or protein. And what you can do is, by a structure-guided engineering process, introduce a so-called hole in the active site of this enzyme by mutation, site-directed, that binds or is complementary to a chemical modification in a so-called bumped substrate. So this is a chemically modified analog of GalNAc, of UDP GalNAc, that has a kind of a chemical diversification as well as a clickable tag. So you can trace glycosylation sites that are modified by this enzyme by using click chemistry. And so the way this works structurally is that you don't change the overall architecture of the enzyme. This is an enzyme that transfers GalNAc. It's not really important what this enzyme does, although it has huge biological function, but this is just kind of a proof of principle to say that you don't change the overall structure of the enzyme. This is an overlay between the wild type enzyme in orange and the engineered enzyme in blue. There's really no change in the architecture, but what you do is you mutate this isoleucine and this leucine in the active site to alanines, and that opens up this hole that is occupied by a chemically modified version of GalNAc. And this is taken from real crystallographic data, so we had a co-crystal structure of this. This is all verified. And what this does to catalysis is that you generate an orthogonal enzyme substrate pair. And that's really cool because the wild type enzyme uses the native UDP sugar, UDP GalNAc, in this in vitro glycosylation experiment. So this is kind of a synthetic peptide with UDP sugar and enzyme. And you monitor turnover. But UDP GalNAc itself is not used by the engineered enzyme. The engineered enzyme, in turn, uses a chemically modified version of UDP GalNAc, in this gray bar here, which in turn is not used by the wild type enzyme. So this is a really orthogonal enzyme substrate pair that's almost perfect in terms of selectivity for different substrates. And so that allows us to kind of look at substrates of individual glycosyltransferase enzymes. But today I wanted to shift a little bit to kind of go a step further into looking into something that we encountered along the way when we wanted to implement this system in the living cell, right? Because ideally you want to kind of know which protein substrates are modified by which glycosyltransferase in the secretory pathway in the Golgi. And so what we wanted to do was to use chemically caged precursors that pass through the membrane and are decaged in the cell. And normally we would want them to be turned over or converted into the corresponding UDP sugar because those are the substrates that are used by these glycosyltransferases. However, if this modification here is too big, the cell doesn't take it up, but it does not enter the native biosynthetic pathway. I hear an echo somehow. Anyway, it's not taken up by the native biosynthetic pathway. And that was a problem for a while until we found a way to bypass that by using two enzymes, a kinase called NHK and a pyrophosphorylase called HX1F283A. And it's not really important what these enzymes do or what they are. In the interest of time, I'm kind of omitting that. If you want to know more about this, please register for the meeting in April. But those two enzymes allow us to bypass this impediment in biosynthesis. So we have an engineered metabolic pathway that lets us biosynthesize this UDP sugar. Right? And so you could say, you need to transfect two more things into a cell in order for this to work. But I'm going to convince you that it's worth it. And so what we can do is we can transfect cells, K562 cells in this case, which are easy to handle, with these biosynthetic enzymes. And we can feed a chemically caged precursor of the sugar, and the cell takes up the sugar and incorporates it into its glycoproteome. And then you can click on a fluorophore onto the living cell. You can wash away non-reactive fluorophore, and then you can lyse your cells and put everything on a gel and image by injafluorescence glycosylation. And so here you can see in this gel, when you transfect the cells with a mock plasmid, with an empty plasmid, there is no signal whatsoever. But when you transfect with these two biosynthetic enzymes, you get a good a dose response curve over 2 orders of magnitude in signal. And so those are all glycoproteins that are modified with this chemically tagged sugar. And so that's great. And then we thought, this lets us implement this bump and hold technology into the living cell, but it lets us also do other things. Like, for example, if we have a sugar that is only used in cells that express an artificial metabolic pathway, this would give us the opportunity for cell-specific glycoproteomics. And so the idea is, a normal cell does not introduce this sugar at all into its glycoprotein. So what if we use a co-culture technique, like, for example, a cancer cell and a bystander cell, such as a fibroblast cell line, which kind of mimics a little bit what happens in the environment of a tumor. And people are dying to know which glycoproteins and which proteins are made by which cell line in the co-culture system and organoids, but also kind of in the living animal, because that lets us kind of define new biomarkers, for example, for cancer. And our technology could, in theory, do that. So we could kind of give cancer cells the opportunity to incorporate these chemical modifications which bystander cells might not have. And so we did that. So we coined this term BOCTAG for bioorthogonal cell-specific tagging of glycoproteins. And what my postdoc Anna Churchill did was to take 41 cancer cells and seed them as a colony on fibroblast cell line that is not transfected. At these 41 cells are transfected with both GFP and our engineered glycosyltransferase transferase of this metabolic pathway. And then you can feed this co-culture system the chemically modified sugar, and then only the cancer cells would incorporate the sugar into the cell surface. And then you can click on a fluorophore and visualize this. And in this image here, you can see DAPI staining, which stains all cells. It shows a lawn of these fibroblasts and a colony of cancer cells, which are GFP positive. And only these cancer cells will have a signal in this BOCTAG channel telling us that we can selectively visualize glycoproteins on these cancer cells. And so just to take this one step further, we can also do this in vivo. And this is kind of the last data point that I'm showing just as a teaser. You can see this also in our bioRxiv submission recently. We took mice and planted tumors that were either mock transfected or non-transfected, or transfected with tumors expressing this biosynthetic pathway. And then you can inject these mice with a chemically modified sugar, but only the cells that are transfected with this biosynthetic pathway would be able to incorporate the sugar into the glycoproteomes. And then you can dissect the tumor and kind of lyse it and click on biotin and put everything on the circavity plot. And then you see that mice treated with only vehicle don't show any signal. Mice that are planted with a mock transfected tumor that cannot incorporate the sugar also don't show any signal. But if they have this biosynthetic pathway, they incorporate the chemically modified sugar into the glycoproteome. And we can also use a positive control sugar that is incorporated into sialic acid, so into all kinds of different glycans in all cells, and that doesn't show any specificity. So we can specifically kind of see which glycoproteins are made by a tumor in an in vivo cancer model. And that lets us do a lot of things. And again, if you want to know more, just join our meeting and you will find out. So I would like to thank everyone who has helped with this. This was mainly led by Anna Churchill and Bea Kaye in my lab, but I thank all the collaborators, funding agencies, and you for your attention.
Thank you, Ben. This was a great first talk and brings us really into the heart of glycoscience here. So our next speaker is going to be Fiona Cuskin. Fiona comes from the University of Newcastle, where she's a lecturer, and her research is focused on understanding the molecular mechanisms of complex carbohydrate degradation in microbial communities. So we are switching from carbohydrate synthesis to degradation, and from mammalian cells to microorganisms. But I feel that the organism is still going to be the same, because these microbial communities in the human gut are probably not that dissimilar from what happens in a mouse gut. I would think, maybe. So this will involve a lot of biochemistry and structural biology. So Fiona, please take us ahead.
Yeah, hi everyone. So thanks for giving me this opportunity to talk about my research, and I'm going to talk a little bit about microbial glycan utilization in the human gut microbiota. So just to give a bit of background, the gut microbiota is emerging as a really important topic, because we know that it's linked with various diseases such as diabetes, IBD, obesity. It's a strange thing, because if you search the gut microbiota and a disease, you will find papers that show that there's links between those, but the mechanistic information about how the gut microbiota works is lacking behind some of that sequence and studies. So it's also really important because it's involved in the development of the immune system, and we know that changes in the microbiota result in allergies and things. So it's quite an interesting and important topic. And from my point of view, if I distill the microbiota down to its simplest sort of relationship with the host, it provides nutrients in the form of non-digestible carbohydrates. So the host through the food that we eat as humans. We provide all of these non-digestible carbohydrates that the microbiota uses. And the microbiota in turn produce short-chain fatty acids and other vitamins, which are absorbed by the host and are beneficial for host health. There's a myriad of other relationships between the microbiota and the host, but for my sort of research, this is the most simple relationship between the two. It's a very competitive environment, and within the microbiota there's two major phyla, which are the Bacteroidetes and the Firmicutes. And the major genus is usually the Bacteroides, and that's the group of bacteria that I'm particularly interested in. And I'm interested in the Bacteroides because they are very good at breaking down complex carbohydrates. So if we look at this figure, what it's shown, it plots the numbers of genes of glycoside hydrolases, GHs, and pectate lyases, PLs, per genome. And the reason why these genes and these enzymes are interesting is because these are the enzymes that are required for carbohydrate degradation. And it plots the numbers of genes per genome compared to the number of different families. And what we see in red is the Bacteroidetes that are highlighted, and they've had a huge expansion in the number of carbohydrate enzymes, degradation enzymes that it has in its genome, and especially the Bacteroides. So if we look at Bacteroides theta iota, which is very well studied, we see that it's got almost 300 genes that are encoding carbohydrate degradative enzymes in upwards of 50 families. Now this is quite old data because it's from 2013, and since then there have been more families and more genes and enzymes that have been discovered and characterized. But what we see is that this group, these Bacteroides, have had a huge expansion and dedicate a huge part of their genome to carbohydrate degradation. So in my group, what we're really interested in is looking at how Bacteroides break down different polysaccharides that are found in the human gut. And Bacteroides are really good to work with because they very nicely co-locate and co-regulate all of the enzymes, binding proteins and transport proteins that are required to break down a particular polysaccharide into a loci. And we call these a polysaccharide utilization loci, or PUL. And for each different kind of polysaccharide, we find that there's a dedicated PUL to break that down. So here what we can see is the typical architecture of a PUL. We have a TOMB-dependent poron, which we call a cis-C, and we have a binding protein on the extracellular surface of the microbe, which is a cis-D. And what we find is that this pair of a cis-C and a cis-D are the fingerprint for a PUL. So we can identify a PUL in a genome because we'll see a cis-C, cis-D pair. And they're really important because they're the transport mechanism for getting polysaccharides or oligosaccharides into the periplasm for degradation by the bacteria and ultimately metabolism for energy. So we have this transport mechanism here. We can also have surface binding proteins which help to recognize and bind the polysaccharide. We have enzymes that degrade the polysaccharide into large oligosaccharides on the surface to bring into the periplasm, where it's further degraded finally into a monosaccharide, which is brought into the cytoplasm for metabolism. They also have a sensor regulator, and the periplasmic domain of this will bind a molecular cue, so part of the carbohydrate, the polysaccharide, it will bind and recognize it, and then the whole system is upregulated so that it can use this polysaccharide as it sees it in the gut. So Bacteroides have numerous different polysaccharide utilization loci, and we're interested in trying to work out how these different PULs work to break down different polysaccharides. There's a huge amount of polysaccharides that are found in the gut. I'm not going to go into a lot of detail, but we get dietary polysaccharides from plants, and we know a lot about how these are broken down by the Bacteroides. You get endogenous host glycans, which include N-linked glycans and O-linked mucins, and we know quite a bit about how these are broken down. But what I'm really interested in are the microbial glycans that are found often as food additives. They're also found in fermented food products and probiotics, and they're also found as normal members of the microbiota. And that's what I'm going to talk about. So microbial glycans in the gut are usually produced by lactic acid bacteria. And these are normal members of the microbiota, Lactobacillus, Bifidobacterium. They often are the dominant members of the infant microbiota. They're found in popular fermented food products. They're used as probiotics, and they produce diverse capsular and exopolysaccharides, and we're interested in how the Bacteroides break those down. And this is a very much an ongoing project in our lab, so I just wanted to show some of our recent data. So the first thing that we were able to do was to extract bacterial EPS from monocultures and from fermented milk products. So you can see here that we've got the microbial cultures, and this is what we end up with at the end, which is just the fine EPS. We then characterized that EPS to look at the monosaccharides to make sure that it was the EPS that we thought it was, and we can see the activities here. And then we also, we've also then started to look at how the Bacteroides break it down. So we can do growth screens where we have a panel of Bacteroides, different strains, and we use the EPS as a single carbon source to see if the Bacteroides can grow on it, then they're able to use it as a sole carbon source. And this is where we got into a little bit of trouble, because when we tried to grow our Bacteroides on these EPS, we found that actually, if you look at the control, this is a minimal media with an EPS from Bifidobacterium animalis. And you can see that the control is very cloudy. It almost looks like it has bacteria in it and it is grown. And that it hasn't. All we've done is added the EPS to that. And this is because those high concentrations we use for growth experiments, the EPS isn't very soluble in the growth media, and it means that we can't measure the difference in OD 600 to look at the growth of the Bacteroides on these EPS. So we were able to do some sort of old school microbiology, if you like, taking time points and doing colony form and unit counts to show that Bacteroides fragilis grew on this particular Bifidobacterium EPS, but that wasn't really what we wanted to do. We wanted a high throughput way to screen all of our different Bacteroides species against all of these different EPS to look for bacteria that grow and use it. So what we thought we would do would be to try and supplement media with a low concentration of this EPS where it doesn't give that cloudy culture, and see if we could see a difference in growth. Now, we knew that Bacteroides fragilis will grow on the EPS from Bifidobacterium animalis. And what we did was we took a rich media, so T by G, so this is very rich. The bacteria will grow on it really nicely. And you can see there the green line that it does. But then if we start supplementing in low concentrations of the EPS, we see that we get an increase in growth rate, which shows us that the Bacteroides are able to use that EPS. Just to show you a control, Bacteroides find Golgi. When we do the same thing, we see that it doesn't use the EPS because we get exactly the same rate of growth no matter how much we increase the EPS. So this gave us at least a way that we could scale up and do some high throughput growths with these EPS to see what grew on which different EPS. What we then went on to do was to take Bacteroides fragilis, and because we knew that it grew really nicely on the Bifidobacterium EPS, we then did some comparative proteomics in order to identify genes or proteins, I should say, that were produced in order to break down the EPS. And then we were able to identify pulls that hopefully are being used for breaking down that Bifido EPS. So we had three different operons that came up. So we had a pull here because we can see we've got our cis-C and cis-D, which are sort of the calling card of a polysaccharide utilization loci, and then it had a few different enzymes in there that are all different carbohydrate active enzyme glycoside hydrolase families. And then we also had two clusters that didn't seem to have a cis-C and a cis-D pair, but we knew from the proteomics that these proteins were present in the culture when we grew it on APS. So the next thing that we did was to clone all of those genes, to express them in E. coli and to try and purify them. So this is sort of the point that we've got to at the moment, where we've got a number of the genes that are cloned, that they're expressing, and we've got some activity. And what's really interesting, and I didn't have too much time to show you all the different activities, but one of the things that is interesting about these activities is the enzymes appear to have activity across a lot of different bifidobacterium EPS. So we have extracted EPS from lots of different strains of bifidobacterium, and we don't actually know the structure of a lot of those EPSs, but what we can see is that the structure has some conservation across different strains of bifidobacterium because some of our enzymes have activity across those different EPS strains. So that's really interesting to us, because if our pull and clusters are upregulated in response to lots of different strains of EPS from lots of different strains of bifidobacterium, that sort of is a different mechanism for carbohydrate degradation, because before this, what we saw is that pulls are very strictly regulated so that they are active on one or maybe two very similar structured polysaccharides, whereas here we're seeing that the pulls may be a lot more promiscuous when it comes to microbial glycans. So we've still got a lot more work to do on this, and like I say, it's very much an ongoing project. But so the final thoughts are that we have shown that Bacteroides are capable of using other bacterial polysaccharides as a nutrient source. One thing we're quite interested in looking at is if the Bacteroides can use EPS from established and grown biofilms, because often these exo and capsular polysaccharides form part of biofilms. And we're also very interested in trying to think about the competitive environment of the gut, and what does the ability to break down other bacterial polysaccharides do for Bacteroides? Is it an advantage? How does it play out in terms of the microbial ecology of the gut and the development of the gut? Because a lot of these strains that we're looking at are from the bifidobacterium, are from infant guts. And we know that there's a development of the microbiota. So how does the ability of Bacteroides to break down bacterial polysaccharides aid in the development of the microbiota from an infant to a mature gut? And then our final question are, in the gut, are good neighbours really good friends? You know, does the ability to break down your neighbour's bacterial capsular polysaccharide really make you a good friend? And that's some of the work that we're doing to look at how that ecology changes. So just to thank the people involved, all of this work was done by Hannah Gibson, who's a PhD student in my lab, and we've got some collaborators who've looked at lactic acid, EPS, and the people in the school and the labs that we're associated with.
Thank you, Fiona. This was a really nice talk, and took us from mammalian systems to bacteria, and now we are going to go back to mammalian systems, but still stay with carbohydrate degradation, but then also move to proteoglycans instead of the type of glycans and polysaccharides that Fiona and Ben have been talking about. So our next speaker is Salvatore Santamaria from Imperial College, is going to talk about proteoglycan proteolysis in vascular and degenerative joint diseases. Salvatore is a British Heart Foundation Basic Science Research Fellow, And the title of his talk will be Molecular Mechanisms Behind ADAMTS Proteoglycanase Activity. Thank you. Salvatore, please go ahead.
Yeah, thanks to the organizer for this kind of opportunity. Can you see my screen?
Yes.
Yeah. So condensation of fat proteoglycans are massive molecules characterized by a number of conjugated sulfate chains. Those are characterized. Those are characterized by diglycuronic acids and N-acetyl degalactosamine. So are difficult molecule to work with because of this massive amount of sugars. The two major conjugated sulfate proteoglycans are aggrecan and versican. So while aggrecan is the predominant proteoglycan in cartilage, versican is predominantly expressed in cardiovascular tissues. So proteoglycans levels are regulated in vivo by protease called ADAMTS. So in the last 20 years, knockout studies have shown that when these ADAMTS activities is abrogated, we see a range of anomalies in a variety of systems. And all these anomalies are characterized by accumulation of proteoglycans, in particular versican and aggrecan. So my work focuses to understand how ADAMTS protease recognize and cleave conjugated sulfate proteoglycans. And looking at versican as a prototype proteoglycan, we try to understand how the different proteases cleave these molecules, which are the substrate binding region or exosites involved, and if the conjugated sulfate chains are strictly necessary for versicanase activity. Finally, we try to understand if there are additional cleavage sites in these proteoglycans. So looking at versican is cleaved by ADAMTS protease at the canonical site, glutamic acid 441, alanine 442, and therefore generates an N-terminal fragment called versicin. So we developed a method which uses an anti-versicin neoepitope antibody that specifically recognizes this cleavage fragment to establish essentially a versican ELISA to measure versicin fragments. So in our lab, we routinely express a full-length versican and its truncated version of V1,5-GAG, which contains only 5 conjugated sulfate chains. So upon digestion with the proteases, with ADAMTS proteases, these proteoglycans generate in this assay almost identical standard curves. And importantly, the assay is specific for cleaved versicin, sorry, for the cleavage fragment for versicin, because it does not recognize the uncleaved fragment. So with this tool in hand, we then started to address the questions that I mentioned before. So initially, we were able to look at cleavage of the truncated V1 fragment, V1,5-GAG, and we were able to, for the first time, to establish Michaelis-Menten kinetics for cleavage of this proteoglycan by ADAMTS5, which is one of the major proteoglycanases in vivo. And as you can see, ADAMTS5 cleaves V1 with 5-GAG with quite high efficiency, with a K-cut of a km in the order of 10 to the 6. And this is approximately 25 more efficient of the closest homologue, which is ADAMTS4. Importantly, we repeated this essentially the same experiment using a full-length V1, and we obtained almost exactly the same results, which was reassuring. And more recently, we also obtained the same data for ADAMTS1, which is one of the closest homologue to ADAMTS5, showing that it's approximately 1,000-fold less efficient than ADAMTS5. And we also confirmed this in a qualitative way using specific Western blot antibodies. So to summarize this first part of the talk, ADAMTS5 looks among the most efficient versicanase, at least in vitro. The second step was to try to identify the exosite involved in this interaction. This is particularly important, especially from a pharmaceutical point of view, if we want to try to manipulate the activity of these enzymes. So the active site of this protease comprise residues, of course, directly involving catalysis, and lies in the metalloproteinase domain. Whereas in the ancillary domains, there are the so-called exosites, which comprise residues involved in binding recognition of the substrate. So if we imagine our versican as a log, and the active site as a saw, we can compare our exocyte to the hands that position correctly the log to be cleaved efficiently by the saw. So they play quite an important role for catalysis. So to identify RTS exocytes, we start to generate C-terminal deletion mutants of the major versicanase that I mentioned before, RTS 5, 4, and 1. And as you can see here, deletion of the C-terminal spacer domain produce a massive decrease in versicanase activity between 10-fold for RTS 1 and 170-fold for RTS 4. While sequential deletion of the cysteine-rich domain and the thrombospondyle-like motif basically abrogate completely versicanase activity. So because the spacer domain is so important, we take a closer look at this domain. So here is a molecular model of this domain, and we can see that it comprise 10 beta strands in a jelly roll topology connected by 9 interconnected loops. And the 3 of these loops make a single interface. So we decide then to swap these loops with those in homologous anti-thin, which is not able to cleave proteoglycans. And this show quite interesting results because essentially 2 of these loops for antis 5 and 4 were shown to be directly involved in versicanase activity. So they do contain exocytes. Importantly, we also show that the same loop are important for cleaving of the other proteoglycan aggregate, showing that these enzymes use the same exocyte to contact both proteoglycans. We also interrogate the structure of antis-5 using a small library of glycoconjugated compounds in collaboration with the University of Pisa. And here is the structure of the lead compound. So these compounds have an arysulfonamide moiety, characterized also by an N-acetylglucosamine moiety. Importantly, they do not contain a Z-binding group to inhibit the active sites. So molecular model show that this inhibitor interact with the disintegrate domain, actually with 2 specific residues, lysines, the disintegrate motif, through the sugar moiety. So these lysines, importantly, are not present in the homologous antis 4, which is the closest enzyme by homology. So when we test this inhibitor for its ability to inhibit these enzymes, we observed that the inhibitor was very effective in inhibiting antis 5, but it did not inhibit antis 4, possibly because those 2 lysines were not present in antis 4. And when we perform side-directed mutagenesis, we observe a decrease in versicanase activity when just one of these lysines was mutated, showing that this lysine may contain an exocyte. Importantly, those mutants were not inhibited by the compound 4B. So to conclude this part of the talk, we show that these positively charged residues comprise a novel exocyte in the anti-5 disintegrate-like domain. And importantly, we show for the first time that glycoconjugated inhibitors can act as exocytes of anti-S proteases. The third question was if the chondroitin sulfate chains are strictly necessary for this activity. And as I showed before, antis-5 cleave full and vertical with quite high catalytic efficiency. When we start removing sequentially sugars from the structure of this molecule, we observe actually that there was or no effect or either actually a slight increase in versicanase activity. Finally, when we abrogate all the chondroitin sulfate chains by truncating this amino acid from the C-terminus, generated this variant called V10 GAG, we did not observe much of a change in versicanase activity compared with the full length, showing that the chondroitin sulfate chains do not seem to be directly involved in the interaction between the enzyme and the proteoglycan in this case. The last question for today's talk is if we can identify additional cleavage sites in chondroitin sulfate proteoglycans, and this part was run in collaboration with Dr. Sunil Apte at Cleveland Clinic. So here I show a brief summary of the experimental approach that we use to identify novel cleavage sites in these proteoglycans. So we express and purify recombinant full-length versican and its fragment, V15 GAG, And then we subject this to digestion with anti-S1, 4, and 5, and their inactive counterparts. Following trips of digestion, we run a mass spec, and then we perform a lab-free peptide quantitation to analyze, to compare the tryptic versus the semi-tryptic peptides of these digestions. And using a statistical test, we were able to identify cleavage, specific cleavage sites. So I report here proteolysis map of V1,5-GAG, the truncated versican fragment, as well as the full length. So we were quite pleased to observe that these proteases were able to cleave very efficiently at the canonical site, glutamic acid 441, alanine 442. But importantly, we show that these enzymes have the distinct ability to cleave at mutually exclusive sites, as we can see here. And most of these events are actually localized in the central glycosylated rich region called GAG beta. We also ask if these events actually occur in vivo, and when we match the peptides that we identified with those present in the peptide atlas database, we observe that some of these peptides were previously identified in large proteomic studies, showing that they may possibly present in vivo. So we are currently trying to map those cleavage events to actual cleavage events occurring in vivo using protoglycan extraction from orthopathic patients. So to summarize, we quite push our knowledge of our proteases recognize the clear conjunctin sulfate protoglycans. We show that these proteases have different efficiency in versicanase activity. They bind these protoglycans mainly with using their spacer domain. And we also show that exocytes seem to comprise positive charge residues, predominantly but not limited to exposed loops. And importantly, although the conjunctin sulfate chains do not seem to be major determinants for ADAMTS recognition, conjunctin sulfate mimetics, such as glycoconjugates, are promising exocyte inhibitors. And finally, we show a new experimental approach that we use to map novel cleavage events in these protoglycans. And I wish to thank all the past and present members of my group, as well as my collaborator in Cleveland, Dr. Sunil Apte, and especially Dan Martin, as well as my collaborators in Pisa, Professor Elisa Nuti, Armando Rossello, and Tiziano Trucinardi. For those of you who want to know more about the novel proteomic method that we put together, we are going to give a talk tomorrow at the UK Protoglycan event. So thanks for your attention.
Thank you, Salvatore. So now we are going to move to the questions. So again, if you have any questions to our speakers, please put them into the questions box on your screen, and I'll pass on to Karen.
Yeah, thank you to all the speakers. The first question that came to the chat was for Ben, and it's from David Jackson. And he says, nice talk. I'm interested in how small a fluorescent tag you can add to a nascent polysaccharide, for example, in the glycocalyx using click chemistry, without disrupting any native interactions between the modified sugar and its interacting partners like receptors. He's interested in the biosynthetic labeling of hyaluronan in immune cell glycocalyx specifically, but any answers that you can provide are great.
So it's a good question. I mean, obviously, kind of a chemical tag is always going to be it's always going to change things one way or another. The question is just how much, and we're kind of teasing this out. I think there are kind of real applications to these things, and we're talking to kind of people like Kathy Murray, for example, in the context of proteoglycans to see what can be done in that regard. I think it's going to be kind of a mix of things. Like it's going to be an impediment for certain things, but not others. I'm not sure if that helps too much. It is going to depend on the particular enzymes that are used for elaboration. But we know, for example, GalNAc analogs are elaborated, so you can make the T antigen disilol T in a cell, and that's maybe that also surprised us. I'm not guaranteeing that this is going to happen the same for every single glycan, but there is some promise in there.
Great, thank you very much. The next question is for Fiona from Jerica Sapotic. Jerica asks, did you observe a lot of difference in EPS composition from different lactic acid bacteria, or are they more or less similar?
We, a lot of the EPS were very similar. So from the different bifidobacterium, we found that the structures were a lot more similar than we expected them to be. From lactobacillus, there was a difference between those and the ones from the bifidobacterium. One of the interesting things that we haven't yet got to look at, just because of time constraints, is the real difference in structure you get from actually what media you grow the bacteria in. So a lot of these bacteria are found in fermented foods or fermented milk products. And if you grow them on a milk medium rather than a sort of lab-based rich medium, you get a different structure. So that's something that we're really interested to start looking at, but obviously we just haven't got around to it yet.
We'll look forward to developments in that area and in the future. Thank you. The next question is for Salvatore. Could you say a little bit more about the role of proteoglycan specifically in the cardiovascular system rather than in joints and lubrication?
Yes, so proteoglycans play a massive role in cardiovascular tissues. Especially my area of interest is especially the aorta. So in the aorta, proteoglycans comprise a major percentage of the media layer. And one of the common things that people have noticed throughout the decades is that aortopathies are characterized by proteoglycans accumulation, and which is incredibly similar to the phenotypes of some of these ADAMTS knockout mice that have been observed before. So the function of these proteoglycans is to maintain the tissues properly wet. So if for some reason they predominate over more strong components of the extracellular matrix, such as the collagens, of course you can imagine that the tissue gets weaker and softer. So this is actually what's occurring, and data from my lab, from our collaborators, have shown that this is partially due to impaired proteoglycanase activity by ADAMTS proteases.
Great, thank you.
Welcome.
Another question for Ben. Which glycosyltransferases are available for the box tag method? Is it only for GalNAc, or are there also available for other sugars?
So for the moment, the GalNAc-Ts are our go-to method. We're working on others, and we have grants to start engineering other glycosyltransferases, but the GalNAc-Ts are kind of our go-to system because it works so well. It works much better than we initially thought, but it's just quite amazing.
Okay, and while we're on this subject, do you see, this is a separate question, did you see any substrate specificity changes when you're doing the bump and hole engineering? Is there a chance that different proteins or protein sites become glycosylated?
Yeah, it's a great question. We checked this initially. We used we were working with Mila Merksic in the US, and we used randomized peptides that were kind of amino acids +1 and -1 positions were randomized, and we saw almost exactly kind of the same profiles between the wild type and the engineered enzymes. So there were no kind of new activities or so coming up, which is a really good sign. And also the structure supports that, so we don't change anything in the substrate profiles.
Can I ask in connection to that? Does the engineered version of the glycosyltransferase result in a different modification of the GalNAc further on in the glycosylation chain? So as you make a larger glycan, does that change if you use the bumping hole one?
I can't tell you that because I mean, we've been mainly using cancer cells for this, just for the sake of being able to see these, right? You know, things like we were looking up to dicellular T just because it's easier to profile these by glycoproteomics. So I can't tell you what happens if you kind of go further down the road. We know, I mean, you know, we know that T synthase takes it, and the ST6 GalNAcs take it, although there might be kind of impaired activity towards those. I think it might also be important to note that, so this is always going to be sort of a discovery tool, right? So, you know, we know the caveats to this. If you kind of, there might be differential recognition downstream, but just in, you know, to see what glycosylation sites are modified by which transferase, I think that's mainly what we're after, in addition to kind of a couple of other things. But, you know, it is cool to see that there is some elaboration.
Okay.
And next, a question for Fiona. How were poles and their substrates initially identified?
Can you just repeat that question? Sorry, I couldn't hear it.
Sorry, how were poles and their substrates initially identified?
So initially, the first pole that was identified was the SUS system, which is the start utilization system in Bacteroides theta at micron. So that's sort of really famously being characterized. But since then, it's a lot of growth conditions where you start looking at does your Bacteroides of choice grow on particular polysaccharides, and initially it was done through gene chip or RNA-seq data. There's a lot of information now in sequencing data that we can start pulling out to look at poles, but yeah, initially it was done through gene chip data and RNA-seq.
Great, thank you. A question now for Salvatore. Are there other post-translational modification of ADAMTS that you think are important for regulating their activity?
Sorry, can you repeat, please, Karen?
Sorry. Are there other post-translational modifications of the ADAMTS that you think might be important for regulation of their activity?
So this is a very interesting question. So if so regarding post-translational regulation, ADAMTS protease at the post-translational level are regulated mainly by inhibition by endogenous inhibitors called TIMs. So those we know that are important for their activity. Another mechanism which we have also quite largely validated is the so-called C-terminal processing. So what happens is that I've shown before that that the ancillary domains contain the exocytes involved in the versicanase and in general activity. But essentially what happens is that those full-length enzymes are quite keen to chop up themselves. So what we hypothesize that it's happening is that because these enzymes are so active, they are active for quite a short period of time, and then they basically there is a feedback loop where they cleave themselves and they remove the exocytes, and therefore they decrease their versicanase activity. We have shown that actually using chondrocyte system, where we show that the endogenous ADAMTS5 is quite C-terminal truncated.
Great. And then the final question for Fiona is, are there special bacteria in the microbiome which break down glucosamine sulfates, like heparin sulfate, and the same or other bacteria families with high sulfates activities? And that's from Rudolf Gruber.
Yeah, so the group of Bacteroides theta at a micron, will break down heparin sulfate and other sulfated polysaccharides in GAGs. It does it quite well. What we find is that if you give pretty much any polysaccharide to the Bacteroides, at least one of them will be able to break it down. But yeah, we've seen, and it's been, I think it was published on a few years back, that they showed it could break down different GAGs and different sulfated sugars.
Thank you very much. this was a really nice session, and thank you to our speakers who have given really excellent talks. Thank you also for the audience for the nice questions, very interesting. So it just remains me to give everyone some final sort of remarks. Firstly, if you are interested in this area, I would really like to urge you to sign up to this meeting that we are organizing from Golgi organization to Glycan function. Any type of Glycan function you're interested in, please come along. We are really interested in hearing what you're working on and feeding it into the discussions. Remember, the abstract deadline is the 7th of February. The Society welcomes suggestions for other topics for these webinars, so please feed that back to the Society. You're more than welcome to contact us as well. And if you have missed any of these webinars, they are also on the Society's YouTube channel. The details are on the screen, I believe now. And you can find out more about these webinars and propose your own webinars on the Biochemical Society webinars website. The next series is going to be on the 3rd of February about computational enzymology, and please come along for that. And please stay connected, join the Biochemical Society. It is really a nice place to find resources, find help with travel grants, for example, and it also is just a good community to be part of. Thank you very much for everyone who attended, and goodbye.